Sorry, I have to clarify just now. This data from this website is the microRNA sequencing data.
Ah, I see. Okay. What about the transcriptome data from, from RNA sequencing platform?
It's from
are
here.
you going to include that also in your research?
Yeah, it is. It's from here. This data is all from here. The sequencing, and then you click on the transcriptomic profiling, and then you get the count, expression count, quantification count.
So is it from RNA sequencing platform? Because microRNA. Yeah. Okay. So like what Dr. Fatma said, maybe in your result you need to justify how many from RNA sequencing platform, how many from microarray, and how many from RT-PCR.
Yeah. Okay. Sure.
So it is possible to do that, right?
Yeah, it is.
Okay. So yeah, another thing that of course you need to have a details on each of data set that you got. Need also to report on that. Beside the platform sequencing, so you need to report what are the informations that important is whether this is melanoma or non-melanoma as well. So
right.
yeah, I think that's all from my side.
I've done better. Yeah, all right, understood.
Yeah, sure.
Okay. All right, thank you, Dr. Nicky. Okay, so I think that's all. Anything to add, Dr. Pasa?
Yeah, I think, Leo, my advice is that you need to work harder to spell out what you did. I know you haven't done this before, but you have good supervisors there to help you. So you need to critically spell out what you will do or what you have done to reach 452 files, so that your results, your finding is structurally understandable and presentable for us to understand. So that's my advice lah for next progress.
All right, sure. So do I need to present all this again on the next progress?
I think Dr. Ong,
Yeah.
I think you just need to follow the advice, make it more structured, so that we can understand better. When I myself, this is also new to a lot of us here, not only me. So you need to talk in a way that we can understand you. Okay? It's a very hard task to make people to understand you, but you have, as a researcher, as a scientist, you need to do that. You need to, this is the the communication skill that you need to learn. How to make people understand what you do, so that you can share it with the whole world, not just say it like that. Okay, I complete this, and then because this and that. Yeah, so that's my advice lah, last from me. I'm looking forward to hear from you soon lah for the next progress.
Yeah, sure. Thanks for the advice.
No need to correct. I think kan that's all. No need to correct. You just need to the next progress lah. Tengok macam mana.
Thanks for the advice. Because
Yeah,
previously I
thank
also
you,
have no
Dr.
idea
Fatma.
on what to present here on data mining. So just getting the data.
Yeah, but Leo, I think both of Dr. Fatma and Dr. Hughes gave you precious advice to go ahead with what you need to do, because when you present it, what we want to see is what have you done, and what do you understand from what you obtain as results, and how do you analyze it scientifically so that you can come out with a conclusion that you can present to us, saying that this is what you have in hand. So I think that kind of information is not transmitted during this presentation. If I be more direct than Dr. Padma. So anyway, I'm not evaluator. Don't worry. So so I think you may want to prepare your next presentation in a more conducive way, so that we can understand what you are doing and why you are doing so, and after you have obtained your results, what type of analysis you have carried out, and what you get it as a result and conclusion, so that we can see okay, it's like that, and it's just like a story of your work. I think that would be nice to you and to all of us too. All right.
Yeah, yeah.
So, okay.
Thank you. Completely
Yeah,
got
thank
the
you.
idea now on what to do on next presentation.
Okay, great. All right. So,
All right.
thank you very much, Leo, and thank you,
Thank you.
Dr. Padma and Dr. Nick Hughes too. I think that we don't have next presentation. Next presentation is later.
Yeah.
Yeah. Rather, is it going to present?
Okay.
Okay.
All right, good afternoon. Thank you. Dr. Ong, are you done?
Yeah, done. Thank you.
All right, thank you, Dr. Ong. So I think that here we come to our last speaker. All right, here I'd like to invite Mr. RSD, okay, voiced by Dr. Shahida. And today there are two evaluators for your project. The first one will be Dr. Kairu, and I will be the second. Okay, without further ado, and Ra'ad, can you please share your slide and then try whether you can move smoothly here?
Okay, Doctor, share now.
Okay, without further ado, here I pass you the floor, Ra'ad. You can start your presentation.
Doctor, you can see slide now?
Yep, can see. Can you turn it to the slide format, please?
You not see slide. Why?
Yep, the presentation mode.
Doctor, me not see slide. Why? Doctor, see slide now?
Yeah, we can see the slide, but this is
But me not see slide.
you can't see the slide.
Okay.
You can see
Good
your slide
evening.
here.
Okay. Good evening. Hello,
Yeah,
everyone.
just do it in the presentation mode.
Okay. Good evening. Hello, everyone. My name is Ra'ad, under supervision Dr. Noor Shahida. My
Excuse me,
progress.
Ra'ad, sorry, I disturbed you. Can you turn your slide into the presentation mode, please?
Doctor, me not see slide. Okay, I'm present now.
Yeah, it's good. Yes, it's good. Please go ahead. Thank you.
Okay, thank you, Doctor. My progress presentation title cloning and expression of the Giardia lambelia recombinant protein SH2 in E. coli. This is my auto line. Introduction. My work related disease, called giardiasis. The giardiasis is caused by protozoa named the Giardia lambelia. The Giardia lambelia is the common agent, diarrhea, dysentery. The Giardia lambelia have two species, the Giardia lambelia and Giardia duodenals. The Giardia have two stages. One stage, trophozoite and cyst. Trophozoite stage is caused infection disease. Cyst the stage transmission disease. My literature view, SH2 in auto properties study, Western blue. Sorry, Victor, I'm not sure slide. Wait. In Orbit study, Western blue analysis identified 21 potential immunogene binding in a trophozoite protein with the SH2 domain contain protein binding and not labeling certificate. The protein was selected for recombinant product in E. coli facility future study on the prevent and roll Giardia. A protein expression in E. coli reduces stable host BL21 for eukaryotic protein expression factor, including barometer to control protein expression in the case BET29C. My research question: how can the recombinant protein SH2 protein product in E. coli and drive from the immunity response to the Giardia lymphoma infection, improve the accuracy and efficiency the Giardia diagnosis method? This is my method. My method, plasmid transformation. I have now a training plasmid transformation in E. coli BL21 expression, collaborate recombinant E. coli in LB21 include with ABTG, optimized expression condition in OD temperature, IBTG concentration, cost including time, purification, the poly SH2 using embolizing metal. I find the chromatography, optimizing purification, barometer, racing types, pure, the condition, organic was protein identification, very protein brought by SDS page, antigen agent evaluation, from Western blue with the serum sample for the infection patient. My result: prepare material, NCL, prepare and storage, LB broth, prepare and centralized storage, IBTG storage solution, prepare, centralized and store in the -20, BS, plasmid stock, prepare and really using kanamycin, prepare and store. Because my plasmid and company now need training, use the different plasmid, the plasmid training, a transformation of LB21 bacteria using the plasmid successful using the kanamycin antibody. I have now two plates. One plate with the bacterial shoe growth after the transformation. The growth plate contain the kanamycin. Negative control shoe, no growth because the bacteria in the agar contain the kanamycin. Result of the training plasmid, the transformation of other bacteria, top 21, the bacteria, top 21, using the plasmid, the successful transformation using the kanamycin antibody, have two plates. One plate, the transformation, the plasmid, other plate, the control, the bacterial shoe growth after the transformation in the agar plate contain kanamycin, negative control shoe, non-growth bacterial, and the agar place kanamycin. This is my conclusion. Experiment clearly demonstration of the principle selective process in genetic transformation. This usage of the kanamycin as selective agent in several the only bacterial have the quickly resistance growth gene can growth. The result support of the hydrothesis. The plasmid contain resistance growth with the successful introduced in the both bacterial strand BL21 and the top 20 event by their growth in keramicin contain agar plate. This look growth in the look of the growth in the negative control available the consent of the selective mechanism and the conformant potential of the kanamycin inhibited the growth of non-transformation bacteria. This is my Gantt chart. This is my reference. Thank you.
All right, Rahat. Okay, thank you. Short and clear. Okay, and
Okay.
so now I like to open the floor. Okay, for any question. Maybe we can start from Dr. Cairo. Do you have any question to run first?
Okay. Good afternoon, Raj.
Good afternoon, Dr. Assalamu alaikum.
Okay. So, Assalamu warahmatullah. Can you hear me?
Yeah.
Okay. So my question, okay. So how you prepare your competent cells? Are you preparing your competent cells yourself, or you are using others' competent cells?
Using the cell.
How you prepare the competent cells?
Can you again, Doctor? Question.
How do you prepare the competent cells?
Competent cells, the transformation?
For the transformation.
Yeah, for the transformation, the
yes.
culture.
How do you prepare the bacteria? For the transformation process.
Yeah. Choose the active colony in my plate. Already have a plate. Have bacteria BL21. Choose the active colony. Culture the colony in the LB broth in incubator in the overnight. After this, check the OD. After this, some bacteria in a new agar. After this, centrifuge, choose the 1 mol in a tube, PCR tube, centrifuge this, put the NLC, wash 3 times, wash by the NaCl. After this, take the plasmid, autoclave in 50 seconds. After this, put in ice. After this, culture a new plate. A new plate have the kanamycin.
That one is a transformation process, right? That one is a transformation process. The one that I asking is, are you preparing your competent cells yourself, or you are using others' competent cells? That you are using your friends' competent cells.
Yeah,
or
your friends.
prepared competent cells.
Doctor, mean LB21?
BL21.
Yeah, BL21 already find in supervisor.
Oh, it means that your supervisor already prepared the competent BL21. How about top 10?
Doctor, now my plasmid in company, I'm used the 2 type of the bacteria. Check my material and optimize the time, optimize my material or the training, because my plasmid is very expensive. I need training and check my material after work in my plasmid.
Understood. But you also need to understand that why you choose BL21 cells and also top 10 cells.
Okay.
So can you
BL21 and the top 10 selected for kanamycin and my plasmid training use the kanamycin. Already my plasmid, original my plasmid, using the antibody kanamycin. Use this bacteria, check my material.
Okay. Understood that.
Thank you, Doctor.
My question, the next question is, what is function of top 10?
The top 10 bacterial.
What is function of top 10?
Yeah. This bacterial function now in my work check the material during transformation. The bacterial input my plasmid. My plasmid encodonic the kanamycin.
You need to discuss, or you need to read more, Ra'ad, because the way that you answer is very limited information.
Doctor, the top 21, not my bacteria. My bacteria LB20.
No,
The top 10.
my question is, because you are using this, you must have information on what you have now. So you are using top 10 cells and also BL21. So my question is, why you choose top 10, and further, why you choose BL21 cells, bacteria?
Yeah. Doctor, this
are
bacteria is available bacteria. And simple growth found more study about this bacteria.
No, that's not the answer. The answer should be the top 10 used for cloning, while BL21DE3 used for expression. That's the answer that you should answer that like that, because you are now lacking of information of the reagents that you use. So if I want to ask you again, PET28C, okay? So can you explain why you choose IPTG to induce your expression or in the PET28C?
Doctor, use IPTG or the kanamycin?
The IPTG. So you must know that what is the PET28C. Yeah. Okay.
Yeah. The IPTG uses the active expression and the fooding the bacteria.
Can you elaborate more on that?
Yeah, the IPTG use the help the reaction and the activation the bacteria and the fooding the bacteria. Same the activation of bacteria and fooding bacteria.
Okay. You also need to know about this. Why is the map of the PET28C? Why is the active sites, or what is the promoter agent that present in the PET28C? So that's why you are using IPTG to induce your whatever genes that you want to express. So I think you still need, even though the information you are lacking, but the result that you are shown here is valid. It's valid because the A is the one that you transform with the plasmid, but the B is a bacteria without the plasmid, right? Yeah. So I just want to suggest that for these results, the A is okay, but the B you should label is to with the, you need to rephrase your label. So it should be control without plasmid.
Yeah.
Okay, because
okay, Doctor,
the
thank you.
way that you label A and B is quite confusing. So I would
Doctor, this first one.
Yeah, I would suggest you relabel the B as a control bacteria without plasmid, while A is bacteria with plasmid.
Inshallah, next time.
Okay. For
Thank you, Doctor.
because this is initial result, so you able to have a growth of your cells after transformation. It means that your transformations work. Okay, but before that, may I know how many colonies that you count for your transformants?
100 microliter.
No. How many colonies that you count for your transformant, successful transformant for one plate? Top 10, right? So can you go for BL21?
Yes. I not check, but I can now check.
BL21, okay.
Yeah.
Okay. How many
Victor,
colonies? How many colonies that
this
you?
colonies is very little. Why? Because I have using 2 microliter for plasmid.
Mm-hmm.
Yeah.
How you make
I don't number. I don't know number.
how you make it spreading like this? Can you tell us how you make it spreading like that?
Spreading using by golf spatula.
Spatula, okay.
Yeah,
So
golf.
yeah, the L shape, the L shape.
Yeah, L shape. Yeah.
Okay, so if we can look here,
Doctor is a little 2 microliter plasmid. Effect show the little growth.
2 microliter plasmid. You mean that you just put 2 microliter of plasmid into your
because this
suspension
plasmid is
bacteria?
tiny. Yeah.
Okay. So if you look at your result for BL21, there is 2 different colonies size.
Yeah. Because different bacteria.
Different bacteria?
Yeah.
No. For BL21.
BL21 and top 10.
For
I use two
BL21,
bacteria.
yes. There are 2. If you look at your A plate, you can see that there are 2 sizes of bacteria colonies.
Yeah.
Yeah. Can you
Doctor.
BL21, don't go for top 10, go for BL21. Yeah, BL21.
Yeah.
BL21, not top 10. Yes, this one. Look at the plate A.
Yeah.
Okay, can you see 2 sizes of colonies there?
The size?
The size.
Yeah.
Yeah.
Doctor.
Can you interpret anything from that? Because you are not discussing the result here, because you just conclude that you get the transformant. Okay.
Yeah.
So any interpretation that you can see from the plate A here? Why you can see two sizes of colonies, bacterial colonies here?
Doctor, because no, I'm using one bacteria, BL21. I think this small colony is first growth.
Okay. I think you need to discuss further with your supervisor. So this is a progress, okay? So like you have some data, but you need to interpret the data. So yes, true that you successfully transform your plasmid into the BL21, the E3, and also top 10. So the information that you need to know is what is function of top 10 and what is function of BL21. The second one is the PET28A, 28C. So you say that you will use IPTG to induce your protein.
Doctor, the infection vector.
Yeah, infection vectors. Yeah, PET28C. Yeah. So the map of PET28C, you need to masterize the map of the PET28C in order to understand more on the expression process. Okay, and the second one is the way that you label of your results. So I think I'm okay. I think I'm done, Dr. Heng. Please.
Thank you, Doctor.
Thank you so much, Dr. Khairul, for your great input. And I hope that Raad, you can jot down all the suggestions given by Dr. Cairo. Very precious, and we also hope that you can discuss further with your supervisor, Dr. Swaida, as she's an expert in this kind of recommended expression. Okay, from my view, actually that I don't have many aspects to ask, but only have a few technical questions. I think part of them already given by Dr. Cairo, and for my one is I like to ask that since that you got this colony and it's so very well compared to your negative control, but how do you ensure that this is the gene that you want? What is the further step you have to do?
The control?
No. A, from the A. Can you see the legend A there?
Yeah. Successful.
Yeah, how do you confirm that this is a particular genes that you want? You have successfully grown into the E. coli. What is the next step you're supposed to do then?
Doctor, after the transformation, culture the colony in plate. The plate have the kanamycin.
Hang on. Because when you clone the gene into your E. coli, sometimes there may some the gene sequence might be be so-called they cannot align, and maybe they're frame shift, and then it will result you cannot get your recombinant protein well to express. So to avoid it, how do you overcome it? Because that you can see your colony so very well here because they all can grow in the kanamycin. Yeah. So how do you ensure that this gene sequence is okay for expression?
Because now I have the blade control. After this, after this I made the expression and check the Western blue and SDS page and Western blue.
Okay. Before the expression, before the Western blot, what is the step you have to do before the expression? How do you ensure that the sequence is okay for expression?
LCMS, check the LCMS.
No, you have to send out for. You have to do the PCR colony and then make sure that the sequence is okay before going for the expression. This one you can check with your supervisor. And okay, other than that, and all right, just that this is a cloning, and another technical is this is just a minor, very minor. You try not to label your wordings too big because sometimes a very small colony will also can be covered by your wording. So this one you have to because pieces of paper they're not allowed for this kind of thing. Okay, the other thing is the other thing is I'd like to know how do you prepare your kanamycin?
The kanamycin.
How do you prepare your kanamycin in the broth?
Go the storage, take the kanamycin, wait the 5 grams, mix with the distilled water. After this filtration 0.20 and 2. After this storage the kanamycin in fridge. The affection of the kanamycin encodonic the bacterial with the plasmid.
So how do we stir out it? How do you stir out the kanamycin?
Yeah, making the kanamycin.
Yeah, how do you stir out the kanamycin?
Use okay, doctor. Use the kanamycin with the first one mixed with the LB agar.
Yeah, how do you stir out it? Because your LB agar is stir out. How do you stir out your kanamycin?
Me making the 200 mol the LB agar mixed with the 200 microliter mixed with the LB agar. After this, make the plate. Make the plate. Now the plate have the kanamycin.
All right.
The kanamycin selective the mycobacteria.
It's okay. My question was, how do you prepare? How do you stir out the kanamycin? I'm not asking about how do you prepare the LB. So check with your supervisor about it. Okay.
Use the kanamycin. Doctor, the use
Yeah,
the
cannibalizing, you need
mixed
to make
with
sure
the-
that it
with
is
the-
stirred up before you want to introduce into the broth to make the agar.
No, first one with the agar. After in transformation mixed with the agar, in expression mixed with the broth.
Sorry. Right. This one just I don't think that, have you put the objective in your slide? Objective? Can I see your
No,
objective?
research question. This is my research question.
Objective.
Doctor, not make objective in line, only research question.
Okay. So next time just put your objective, because I still remember that your work basically is aimed to produce the SSO protein, right?
Okay.
Yeah. I just want to make sure that this is in a very early part. I think that you already achieved to a certain percentage. So doesn't matter. I think that I have nothing much to ask, but just a few technical questions and some suggestions given by Dr. Cairo and myself also give you some want to test your understanding. But try
Dr.
to
Hang,
understand about the background of this cloning. It looks like quite easy and simple, just because some of your senior have done it before, and you just follow the step, but you have to learn it out because there are quite a lot of the challenges in the cloning as well. So I think that I have nothing much to ask Ra'ad, but what I can do is try to jot down about all the questions given by the evaluators, then try to improve for your coming project and the presentation. All right. So right now, apart from myself, is there any question from the floors?
Dr. Heng,
Yes.
for research question here, I think Ra'ad need to look again, because his project is more towards expressions of the recombinant protein, the expression as purification. So it's not go for improve the accuracy and efficiency. So the question
Okay,
should be, the question need to be rephrased or need to be rechecked again, because it's not
Okay.
resemble to the objective of
Okay.
your
Okay,
So
thank you.
study.
okay, please take down the
Okay,
note,
okay,
okay,
doctor.
just to rephrase sensitivity, specificity, and the sensitivity, I think. Okay, thank you,
Yeah.
Dr. Cairo.
Okay,
Is there
doctor.
any other questions? If no, and thank you so much, Ra'ad. So I wish you good luck
Thank you,
and all the
doctor.
best
Thank you,
for
Dr. Cairo.
your
Thank you, Dr. Shahida.
future works. So I'm here to start presentation for Ra'ad. Thank you so much. Thank you.
Thank you so much, doctor.
们搞完了吗？他们搞完了吗？这个会议关闭了。哦，我也觉得他们感觉明天，我不知道，我也没听。那搞完了，发书，应该是1个下午。不知道，搞不清楚。应该是下午1个，我记得大叔的这个会议。然后孙亚文的是廖还是谁忘了？没有啊，我没听啊。这样吗？我为什么看到苏珊娜的那个投屏了？这他投屏失败了，我不知道，搞不清楚。哎，土香哥有那个免费的外带的东西吗？这个盒子好贵啊，花了我2马币，有病！我操！那下午还去不去啊？这么快呢？你什么回速度？这这合着他们一个人20分钟，那424吧，那个中间停1个小时，那不一个人20分钟？也没人理我，我是现在在干嘛？土香哥有那种免费的袋子吗？外带的。